Fantastic.
Wonderful.
All right.
Wonderful.
That's it. All right. Thank you so much, everyone, for being here today. We're super excited to welcome you to our second year of the Resilient Brain Current Research Landscape panel, focusing on traumatic brain injury developments in the field within research and the clinical setting. And we are super excited to welcome 3 wonderful panelists today. Before we dive straight in, I would like to introduce Sharik Jamani, who is the founder and president of Columbia Synapse, to share a few brief words about our organization's mission. So Sharik, I'll pass it over to you.
Thank you, Chinmay. Great. Fantastic. Thanks, Chinmay, for the introduction. So just for as Chinmay mentioned, wanted to briefly introduce Columbia Synapse and how this group grew to be what it is right now. So we're a New York City-based group governed by principles of community building, advocacy, and obviously neuroscience, which is why you're all here right now. So we draw from a wide network of resources, and it's been a privilege to interact with different hospital systems to organize a diverse portfolio of programs that promote a greater social and a neuroscientific understanding of brain injury, not only at Columbia, but also in New York City. Next slide. So to that end, our programs primarily hinge on 3 principles: neuroscience, community, and advocacy. And this event in particular, it's the second year right now. It hinges on neuroscience, very obviously, as we have wonderful speakers who will be sharing their research, clinical research, and neuroscience research related to brain injury. But also we host community programs, including a peer support group and a volunteer buddy program. And at the helm of all of this is just our mission of advocacy, because we try to leverage human capital, and try to promote an understanding of what brain injury is for the population around us. Next. And really excited for this event to kick off very soon. I'll be handing it off to Chinmay. But before I do, I want to invite you to stay connected with us through our website, our Facebook, Twitter, email, or Instagram. And next slide. And if you'd like to join our mailing list, just receive information about upcoming events similar to this, and our conference in the spring, and our quarterly newsletter, you can sign up on this following link, the tinyurl.com/Columbia Synapse. And with that, I'm going to hand it over to Chinmay, who will introduce the speakers for today.
Thank you very much, Sharik. As Sharik mentioned, my name is Chinmay Bolusu. I am one of the co-vice presidents of events for Columbia Synapse, alongside my peer Ling Hao, who's also here today. Ling Hao, if you want to say hello in the chat. Just a quick notice before we get started with our wonderful speaker introductions and presentations. This event is being recorded and will be posted on the Synapse YouTube channel afterwards. So if you do not wish to be in the recording for your likeness or your voice to be in, please turn your video off and stay on mute if you so desire. And as a quick note about the structure of the event, we will, after these quick speaker introductions, we will be diving into presentations from Dr. Koliatsos, then Dr. Bergold, and then Dr. Sacks-Zimmerman. Afterwards, around 4 p.m. Eastern Time, New York City time, we will be starting our Q&A discussion, part of the panel, where our events team member, Kosi, if you want to wave to everyone, Kosi will be moderating our panel and posing questions to the speakers. And during this time, if you have any questions that pop up, we would kindly request that you please message either me, my name is currently listed as Columbia Synapse, or Ling Hao Kong, who will be guiding our questions to Kosi to ask during the panel as well. So just to keep the chat clear and to avoid spamming the chat room, we would kindly request that you please message either Columbia Synapse, which is me, or Ling Hao, and we will be sure to list your questions as well. With that, I wanted to thank you once again for attending. And here we go with our wonderful speaker introductions. Thank you so much. First of all is Dr. Koliatsos, who is a professor of pathology, neurology, and psychiatry and behavioral sciences at the Johns Hopkins University School of Medicine. His primary research interest is the mechanisms of traumatic and degenerative brain injury and repair, focusing on cellular therapies for neurodegenerative diseases. He earned his medical degree from the University of Athens Medical School in Greece, and later completed a residency in internal medicine and neurology at Crete Naval Hospital. a psychiatry fellowship at the University of Athens Medical School, and a neurology fellowship at the Johns Hopkins Hospital. He also completed an internship in internal medicine at Franklin Square Hospital, followed by a residency in psychiatry at Shepherd Pratt Hospital. He currently serves on the faculty of the PhD graduate program of Pathobiology of Disease and leads the TBI Research Center in the Department of Pathology. As a clinical neuropsychiatrist, he also sees patients with TBI and degenerative dementias. And thank you so much, Dr. Koliatsos. It is wonderful to have us with you today. Our second speaker is Dr. Peter Bergold, who is a professor of physiology, pharmacology, and neurology at SUNY Downstate Health Sciences University, where he joined the faculty in 1991. He received his Bachelor's of Science in Biology from Trinity College in 1977 and a PhD in molecular biology from Cornell Medical College in 1986. Dr. Bergold completed his postdoctoral fellowship in neuroscience at Columbia University and has been a member of the National Neurotrauma Society since January of 2007. Professor Bergold's specialty is in neuropharmacology, with a focus on traumatic brain injury and the development of drug combinations to treat traumatic brain injuries. Thank you so much, Dr. Bergold, for being with us today as well. And last but not least is Dr. Amanda Sacks-Zimmerman, who is an assistant professor at Weill Cornell Medicine's Brain and Spine Center. After receiving her undergrad degree from the George Washington University and her PhD in clinical psychology from Fairleigh Dickinson University, she went on to complete 2 postdoctoral fellowships. Her first was at University Behavioral Healthcare, UMDNJ, where she studied neuropsychology, and where she procured research projects examining cognitive correlates of emotion in dementia. In her second fellowship at Mount Sinai Medical Center, she assisted the making of a manualized cognitive behavioral treatment for individuals post-TBI. Before joining Weill Cornell, she was a faculty-appointed member to the Department of Anesthesiology at NYU Langone Medical Center's Rusk Institute of Rehabilitation Medicine. Here, she managed research for cognitive issues in post-op cardiac patients and related to compliance in the wound care population. All in all, she is a board-certified neuropsychologist with extensive research experience in the world of cognitive impact on brain injury and remediation programs. A huge thank you, Dr. Sacks-Zimmerman, and to all of our wonderful speakers today. We are super excited to hear about your research and current work and gain insight from you. And with that, Dr. Koliatsos, I would like to pass it on to you for our first presentation.
Thank you so much, Chinmay. Can you all hear me?
Yes, we can.
Thank you so much, and thanks for using a picture of mine that's about 10 years old. That's terrific. That's wonderful. So I'm going to be talking about what I've been doing in the laboratory in the last several years. And this is a white matter. I've always studied axons in my career, and I'm trying to apply some of this knowledge in studying traumatic brain injury. And I will focus on some new therapeutic opportunities. So I'm going to be talking about axons and white matter, and I'm going to talk about some of the new therapeutic opportunities, at least the way we see them in the lab. That's a little bit of a departure from previous work of mine that was based on neurotrophins and stem cells. But you can certainly ask me about that work as well in the end, because I saw a related question. So as you all know, neurons are peculiar cells that really outlay themselves into their axons. Neurons, as opposed to liver cells or other glandular cells, are their axons. The nervous system is a set of axons. So I like to use this example that if you take a motor neuron of the anterior horn or a large DRG neuron, if the cell body is about 5, say, micrometers diameter, the axonal outlay is about 1 meter. So if you know anything about Baltimore, and this is the size of an orange sitting at Johns Hopkins, the terminal is down at the Inner Harbor. That's pretty remarkable. And this really has led to a progressive independence of the axons from the cell body with a different set of molecular determinants of survival and degeneration. Now, if you take into account the entire set of branches of axons and terminals, this is becoming a formidable ratio of about 30 microns to 100 meters in the case of a cholinergic neuron of the basal forebrain, for example. So this is a remarkable biological fact. This is a very important part of the hypothesis I recently set as the background of research in the laboratory. Now, a second important fact is that axons and white matter, and particularly important for the human brain, as you ascend the phylogenetic line, this is a rat brain, this is a sheep brain, and this is a human brain. For every unit you gain in gray matter, the gain in white matter is exponential. This exponent is larger than 1. So for every unit of volume you gain of this, you gain much more of this. So this is particularly important at the same time. It really exposed the white matter to a set of risks that span the entire spectrum of exogenous injuries, from mechanical trauma to hypoxia ischemia. And this is how these different injuries look like under the microscope. These are large axons that obviously are quite distorted. They're wavy, they're undulated, as we say, and they have this classical terminal swellings after a motor vehicle accident. This is from the frontal lobe of a motor vehicle accident victim. This is someone actually who has exposure to blast. The axons are a little different. They're probably smaller. The configurations look a little different. So the neuropathology signature is a little different. And this is actually the brain, the frontal lobe white matter of someone who died from opioid overdose. And we see increasingly more and more of these cases because of the unfortunate epidemic that's plaguing America, but also other Western countries. So there is a across the board vulnerability of axons and white matter that perhaps is of a different nature here than here, but there are quite a few common denominators. Now, in the most classical case of mechanical injury to axons, what we call diffuse axonal injury, this can be actually a pretty serious issue with multiple punctate bleeding sites that also correspond to sites of actually groups of axons that are disconnected, and where the angular acceleration forces are particularly severe. You see actually a dislodgement of white from gray matter in what we call gliding contusions. Obviously, many people die, but some people survive. This is actually happens to be a patient of mine who's a 24-year-old woman, the victim of a motor vehicle accident, who was ejected from a car. This is actually a pretty typical scenario, and sustained diffuse axonal injury. This is 6 months post-TBI, and this is a memorized sequence that we call susceptibility-weighted imaging. This is very good in detecting heme as, pitch black sites. And you see the diffuse nature of these lesions. They're here, which is in superior cortical white matter junction. But you see some here. You see quite a bit in the basal ganglia, and then as you go into the basal forebrain and the brainstem. So although many people die, some survive to see the outcome of this, which in many cases is not particularly benign, because some of these patients with progressive time will have atrophy of the brain. This is another young person, 1.5 years, not 6 months like the previous one. And here you see actually evidence of diffuse axonal injury, in this case in a FLAIR scan. In T1, you also see this gliding contusion. And what you notice here in the mid-sagittal section of the brain is the atrophy of the corpus callosum that really spells disaster in really bihemispheric connections of this forebrain. So the long-term consequences, but also the behavioral, cognitive, and mental consequences in many cases are pretty severe. So the way we understand axonal pathology for about 100 years from the days of Nissl and Waller is that if an axon actually has an injury here, the cell body is undergoing some adaptive changes known as chromatolysis, with a very intense transcription translation profile that manufactures new protein and ships it so that the axon can be repaired in the PNS. And the axon actually is fragmented in a very orderly way that's known as Wallerian degeneration, these classical ovoids that prepare the axon for repair because also Schwann cells come in and create these orderly columns known as Bigner bands so that the PNS can repair itself. CNS is a different story because CNS cannot repair itself as well. And the Wallerian degeneration has some significant, permutations of that theme. Now, fast forward in the last few years, we understand that a key executive signal for this, which really involves this part of the axon, we call this the Nissl domain and this the Waller or Wallerian domain, is actually a tall interleukin-1 receptor. This is actually a very interesting family, superfamily of receptors that are very ancient and highly conserved. In fact, there's some versions of this in plants. But this is one of the most ancient of these ancient receptors that has an autoinhibitory domain, it has a multimerization domain, the sterile alpha motif domain, and then the TIR domain that we now understand acts as an enzyme that digests NAD. It's a NADase, and I will get back to this in 5 minutes. So we're very interested in this in the laboratory. Another thing which we found, and that's very important discovery for us, is that if you take a model of traumatic axonal injury that we use in the laboratory, and this is an angular or rotational acceleration paradigm based on the model of Marmoro, where we use impact to accelerate, you see the whiplash in this mouse. So the head goes down, and then it bounces back up in this whiplash scenario. We're talking about an acceleration of about 4 meters per second. You see many axons, for example, in the optic nerve injured, and you see that a majority of the cell bodies of these axons actually bear the brunt of injury response, yet only a subpopulation dies. So the injury response is much larger or wider than the degenerative response. And this led us think that after, say, mechanical trauma, axons get injured. Some of them can advance to become completely disconnected and undergo classical Wallerian degeneration, but some of them may come back to be repaired. What we therefore need to do is try to intervene in the best way we can and bias this dynamic system. It's a selective system, it's not an instructive system, to really go back to this instead of going back to this. So our game here, our therapeutic target, is these axons that are potentially repairable. As long as they do not turn on Wallerian degeneration molecular signals. So if we can intervene in these signals, we may actually preserve an otherwise rescuable axon. And that's really the gist of our laboratory hypothesis. Now, our work down at the biochemical molecular level concentrates on NAD, nicotinamide, adenine, dinucleotide. This is a universal cofactor in redox reactions that's generated by B3 nicotinoyl precursors in some very well-defined biochemical pathways and de novo. Disturbances in these pathways, either dietary polar amino acid transport disorders like Hartnup disease, or actually B3 deficiency, can cause severe deficiencies in NAD that have a number of neuropsychiatric consequences, including neuropathy. The reason why this is the center of our biochemical world is that one of the first things that happens that happen in an axon after transection is the deprivation of this key enzyme that synthesizes NAD in most of the salvage pathways, the NMNAT1 to 3. This is an important event because it actually obviously causes NAD deprivation in the axon, and we think this is a key factor. At the same time, we're going to focus a little bit on this, which is the precursor of NAD. Now, let's really see what happens in a normal axon. Let's focus on this particular equation. A normal axon, say the nicotinamide recycling pathway, you create NAD through the intermediate that is the nicotinamide mononucleotide, or NMN. That's normal. Now, if you have an injury, you don't have this enzyme. Actually, some of this is degraded via well-understood molecular pathways, and what you have is an accumulation of NMN and obviously disappearance of NAD. And in the last several years, we also know that a further reduction in NAD is happening through the action of this enzyme. I talked about this enzyme that acts as NADase. So essentially, the axon is totally depleted of NAD. This is the injury scenario. To explore the possibility of this scenario actually operating in mechanical trauma, we've set up a number of systems in the lab, in vitro and in vivo systems that we try to upgrade to high resolution. One is a microfluidic system where we separate cell bodies from axons so we can study them independently. In fact, we separate axons in 3 domains so we can distinguish between proximal and distal axon. We can cut the axons with a microknife or with a laser beam, or we can actually compress them with a particular device. And we follow the process after a few hours. This is before injury, how these axons actually intertwine in this compartment. And this is actually classical Wallerian degeneration. This is how it looks actually. This is, you're gonna see these axons. This is where the cut is. These are axons in these little channels here. And you see the Wallerian degeneration, this actually almost explosive fragmentation in multiple little domains. It happens throughout the length of the axon in actually one little click of a finger. This is how it looks. It's a high resolution model where we can make some important observations. And another high resolution model that we really
join the meeting
is the in vivo clarity preparation. This, you see a clarity preparation of the mouse brainstem, and this is the corticospinal pathway, and you see how concussion is all these dots are actually axonal swellings and Wallerian profiles coming down from the reticulospinal tract. So this is a high resolution profiles that allows us a 3D reconstruction, but also a very accurate account of the totality of these lesions. Other high resolution strategies are with high, intensity fluorescent microscopes using similar techniques where we can actually see details of individual axons. So in the injury scenario, we lose NAD, we have accumulation of NMN. What can we do about it if we're going to think therapeutically? We lose NAD, we gain in the precursor NMN, and obviously we have activation of SARM1. These are the 3 key facts. What can we do therapeutically? One is to take out the gene that's the executor of Wallerian degeneration. And another way to do it that we're particularly interested in is actually block this enzyme. And initially, we didn't know what this blockade of the enzyme is going to do to the executor of Wallerian degeneration. Now we learn more, and I'm going to tell you a few things before I conclude. So if you block NAMPT, the effect is quite intense. This is how axons would normally degenerate. This is the generated index. This is 80% of the axons that really are gone by 48 hours after transaction. Very clearly, the NAMPT inhibitor FK866 rescues many of these axons, particularly early on, although later on, the trend is for them to reach essentially vehicle levels. But if you block with knockout the SARM1 enzyme, you almost rescue quite a bit in a very prolonged period of time. If you add FK to this, you don't really gain any more, which means that the rescue effect is driven by the gene. But still, you have a significant effect of NAMPT inhibition. If you choose a number, if you select a number of NAMPT inhibitors and do the same experiment, they all migrate this curve in the same way, clearly telling you that at least pharmacologically, NAMPT is a legitimate target for intervention. However, you need to be careful about windows because although these drugs work well if you give them between 2 and 4 hours, they don't work as well if you give them right from the beginning or if you give them very late. So there's a time window of efficacy that is important mechanistically, but is also very important therapeutically. So NAMPT is a target. The effect is similar, but not exactly identical to knocking out SARM1 gene, and the effect has a time window. If you look at this time window with some metabolic detail, looking at exploring the key players here, you see that between 2 and 4 hours, a couple of things happen that are important. One is the acute increase in this metabolite that shows SARM activation. And the other is also a very significant increase in the precursor of NAD, which is NMN. So these are the two key events we're talking about, the activation of SARM1 and the accumulation of NMN. Do the two have any relationship with each other? Why, for example, if you block NMN, you would also have an effect of SARM1 if you were to think therapeutically? An answer is that in the same time window that you see efficacy of NAMPT inhibitors, you also see the deleterious effect of NMN itself. So you see that the effect of NMN here at 2 and 4 hours is pretty significant. Whereas if you really give it, say at 0 hours, you see absolutely nothing. So here you have a defined time window where if you block NAMPT, you see an effect. And if you replace the product of NAMPT, you negate the effect. What does it have to do with the activation of SARM1? We're actually trying to find out. We are doing similar, we have done similar experiments in vivo using these high resolution models. This is actually how concussion is in the corticospinal tract in a mouse that has a full complement of SARM1 gene. And this is how it looks in a mouse that does not have SARM1. You see the difference in numbers of these swelling profiles of diffuse axonal injury, and the actually counts bear this out in various domains of these corticospinal tracts. So we have a very significant in vivo evidence as well to corroborate our in vitro evidence that SARM1 knockout and also NAMPT inhibition. We have some data with NAMPT inhibition that I don't have the time to go over. Finally, we're very interested in another very important pathway that's interacting with SARM1, that is the universal MAPK injury signal cascade, which links this current work with my previous work on trophic factors. This is actually the pathway that gets turned on if you deprive sympathetic neurons of NGF. So this is how NGF deprivation or neurotrophin deprivation kills cell bodies, by activating this MAPK injury kinase cascade, the MAPK kinase cascade. But this also interacts, this mostly affects the cell body, but it also interacts with axonal processes because this further digests NMNAT2 that is depleted in the axon. So you have a passive depletion, and on top of that, you have a digestion of a version of this enzyme with the downstream products of the MAPK cascade. And fair enough. If you add inhibitors of this pathway to FK, in a way you block both pathways, the gains are very, very significant. You see what happens here with if you combine FK866, a NAMPT inhibitor, with GNE at the time that's optimal for inhibition. It's here. And you see what happens if you really do not use the GNE. If you say take it at about 12 hours, you see that you're about 20% degeneration index, where this is clearly above 20%. So this is currently what we're doing. We're very much interested in the problem of diffuse axonal injury because we think it's a universal underlying neuropathology in trauma, but also it's a common denominator with other kinds of injurious processes, including hypoxic ischemic encephalopathy, but also it's common in glaucoma and peripheral neuropathies. We believe it's worth exploring in the case of traumatic brain injury because we think it is a highly conserved mechanism. So we're talking about a very important biological phenomenon, much like apoptosis, that has clear molecular players that if we understand a little better, we can tackle both with genetic molecular approaches, but also with small molecules. And currently using high resolution in vitro and in vivo models, we're focusing on the SARM1 pathway as it interacts with the salvage pathways for NAB synthesis, NAD synthesis, and also the interaction of this pathway with the universal injury signaling MAPK cascade. And based on this, we designed molecular and pharmacological research strategies. And this is really where we are at this juncture. Thank you so much. Obviously, this is a work that's involved many people. I'd like to mention specifically the work of Thanasis Alexandris, Nick Yogas, Jiwon Ryu, Derek Wellesby, Don Zach, and a whole host of other people who've helped me through my basic science and research work through the years, and all the funders of this work, to which we are deeply grateful. Thank you very much.
Thank you very much, Dr. Koliatsos. If everyone in the audience could give some virtual applause if on video, or some Zoom applause as well. Thank you very much. Wonderful presentation. Next, it would be wonderful, Dr. Bergold, if you could share your slides and share your work with us. Dr. Koliatsos, would you mind unsharing your screen, please? Thank you.
Now. And I'm just going to make sure, am I muted or not?
We can hear you, you are unmuted.
Okay, so thank you very much. And I'm actually on the wrong slide, so let me go back.
Oh, Doctor, we're actually unable to see your slides at the moment, by the way.
You cannot see my slides.
No.
Let's see. Okay. Maybe it's this. Oh, wait a second. This does not look good at all. Let's see. I apologize for this.
Or if you believe it'd be easier, I'd also be happy to share my screen. If you don't mind.
Let's see how this works.
Oh, wonderful. We can see.
Okay. So, first of all, thank you very much for the opportunity to talk with you today. And also, I'm very impressed about the size of the meeting, considering that, at least in New York, it's probably the nicest day we've had in weeks, and also of the very monumentous events that have occurred today. So anyway, thank you for having me. Let me just mention a few things about the work that we're doing on traumatic brain injury. I mean, it's a very serious health issue, with 1.4 million people in America each year sustaining head injury. The major form is this acceleration-deacceleration injury. in which it's the movement of the head. And this is something that Dr. Koliatsos showed with his model and something I will show as well. But also the injury can also be a direct impact onto the skull, which can cause a contusion. So both of these contribute to traumatic brain injury. So one of the major problems that we have is that there are no drugs. And one of the reasons is because of the heterogeneity of traumatic brain injury. I mean, first of all, the head is not injured in a uniform way in the clinic. And also a variety of different agents can induce traumatic brain injury. There can be blunt impact, there can be penetrating traumatic brain injury, there can be blast. But I think the real problem, and this is again something that Dr. Koliatsos brought up slightly, is that the TBI evolves very rapidly. The damage to the brain begins within seconds, and it has a very complex and evolving pathophysiology. Only recently have we started developing objective endpoints. So now basically, if you want to do a clinical trial for virtually any neurological drug, you have to use some type of objective measure to show that there's an improvement. You just can't simply say to the patient, are you feeling better? Can you go back to work? This sort of thing. But I'm going to make the argument also, as I think because of the complex pathophysiology of TBI, I think it's going to be very difficult to treat it with monotherapy because inhibition of one pathway or inhibition of one component of it may be insufficient to give you a true therapeutic effect. And so this gives you, in a very, very cursory way, some of the events that are occurring during TBI. Within moments after injury, there's necrosis, there's the axonal injury that Dr. Koliatsos mentions. There's also programmed cell death. There's large-scale demyelination. And there's also substantial, and I think very important, neuroinflammation. And what we're becoming even increasingly aware of now is that a TBI, which was initially seen as a relatively acute disease, now clearly has chronic components. Again, this was mentioned by Dr. Koliatsos, and one example of it is the ongoing degeneration of both gray and white matter that we're seeing mostly in human MRI studies. So, again, there's something that was mentioned in the earlier talk. I mean, a key thing is the therapeutic time window. I mean, one of the things that we have found in a large number of preclinical studies of TBI is lots and lots of things work if given either before the injury, which is clinically of no use whatsoever, or even within an hour or two after injury, which I would argue, again, has very little value. I mean, the two most recent clinical trials for moderate to severe TBI, which were the PROTECT-2 trial and Synapse trial, which looked at progesterone, they couldn't treat patients. The fastest they could treat patients in the PROTECT-2 trial was 3.6 hours post-injury, and in the Synapse trial was 7 hours. And even then, they were turning away a third to a half of the patients. So we're just, since we don't know what it is, in my laboratory, we're basically saying, I think if we have a drug that works, that we can dose 12 hours after injury, we could capture many of the patients who have moderate to severe TBI. But the problem is that the majority of TBI, in fact, is mild TBI or concussion. And there's very little clinical data about what the time window of that is. And patients with mild TBI typically don't seek treatment until their symptoms persist, their headache persists, their dizziness persists. And so in the laboratory, we're making the working assumption that if we could have a drug that we could treat 3 days after injury, we could capture a lot of these mild TBI patients. And so approximately more than 10 years ago, I got a grant from the Army to basically screen FDA-approved drugs in combination and to see if I could find drugs that worked synergistically. And just to remind you what synergy is that the combination of the drugs, the therapeutic effect is greater than the sum of the therapeutic effect of the individual drugs. And we screened many drugs. And the combination that showed the most efficacy, and I'll talk a little bit more about the screening in a moment, was a drug called minocycline. And one reason I picked minocycline is because, it has multiple therapeutic effects. It's a very potent anti-inflammatory, it's an anti-apoptotic drug, and it's an antioxidant drug. And also, most importantly, it was very clear that this drug entered the brain, because this drug was actually originally developed as a synthetic tetracycline to treat bacterial meningitis. So we knew that this drug could enter the brain even with an intact blood-brain barrier. The second drug was N-acetylcysteine. That again, this is of many drugs. It is shockingly safe. And one of the things about drug development is safety is actually more important than efficacy. A lot of drugs go down not because they aren't effective, it's because they can't be dosed in a safe way. N-acetylcysteine you can buy in a health food store. It's over the counter. It's a pretty good anti-inflammatory. It's an extraordinarily good antioxidant. And so one of the things that was very useful is, again, these are sort of different actions, and that's why we were hoping to find synergy. But interestingly, it's also a very potent modulator of the neurotransmitter glutamate, which is the excitatory neurotransmitter of the brain. And as a result, this is now in dozens of clinical trials for every neurological disease you could imagine. And the results so far, unfortunately, have not been very good as a single drug. So we wanted to use a model of traumatic brain injury. Again, not unlike what Dr. Koliatsos mentioned. We want to see what defines TBI in my mind is the movement of the head. And so we modified a TBI method using a pneumatic piston. And in this case, we strike a mouse's head on a foam pad where you can see movement of the head. And one advantage of this, and this again was mentioned by Dr. Koliatsos, is this produces a disproportionately large amount of white matter injury. And so this is just an example of what the gross anatomy of these brains look like. You can see that we're hitting, we're striking the parietal lobe. There is some deformation of the skull, so we do get a contusion, but we also get injury due to acceleration, deacceleration. And so one of the things that we did 10 years ago is one of my colleagues at Downstate, Dr. Andre Fenton, who's now at NYU, had developed this very, very powerful behavioral test. And the one advantage of this behavioral test is animals could learn it in a matter of a few hours. And so we wanted to see, I was very interested in whether drugs could give you a therapeutic effect on this test. And so this is just a movie of what the test is. It's called the active place avoidance. Now this is speeded up 8 times, but basically the animal is on a rotating arena that's going 1 RPM, and it's learning to avoid a computer-generated shock zone, which is sort of that red V-shaped region. And if you notice, the animal has gotten very good at avoiding the shock zone. This animal could learn that again in a matter of a few hours. The thing that's most striking about this task is it's actually quite difficult, because normally when a mouse is placed on an arena like this, it deposits feces and urine on the arena and uses olfactory cues to navigate. And if you notice, because the arena is rotating, the olfactory cues, in fact, are not informative. And so the animal has to ignore the olfactory cues, which is its major sensory modality, and attend to visual cues in the room. And the thing about this task that's so interesting is, and again, this is some work by myself and some work by Dr. Fenton, is this is one of the few behavioral tasks that require both 2 copies of a brain region, the hippocampus, and most very interestingly, also requires the white matter connections between the 2 hippocampi. And if these things are intact, the animal can learn the task. And what we found was, is that we could dose this drug combination at 12 hours after injury and look at the number of entrances into the shock zone. So these are animals that are treated with saline. Again, this is 4 trials. So in 4 trials, the animals are hitting asymptote on this test. So, and this is an animal that is, this is an injured animal. This is an animal treated with saline. Excuse me, this is an injured animal treated with saline. This is a sham animal. And this is our animals treated with the drugs. And so to make a very long story short, I can show you quite a bit of evidence that we're both, by dosing these drugs at 12 hours, we're both restoring function to the hippocampus, as well as the synaptic connections between the 2 hippocampi. Now, if we dose the drug at the first dose of the drug at 24 hours, we lose this therapeutic effect completely. So we use another behavioral test in my laboratory called the Barnes maze. And so again, I'll show you a video of it. In this case, a mouse is placed on a brightly lit arena, and around the periphery of the arena are a variety of holes. And one of the holes contains an escape box. And the animal has to learn to escape the Barnes maze. And they do that because they don't want to be in a brightly lit open region. And unlike the active place avoidance task, which I previously showed you, Barnes maze can be acquired with 1 hippocampus, and it does not require the white matter tracks between the 2 hippocampi. And what we've shown here is, this is now the acquisition of Barnes maze. And Barnes maze is actually a much more difficult task for the animal to acquire. And so this is this would be a sham animal, and this is an injured animal. And this is the learning curve of an animal that was first treated 3 days after the injury. And you can see that we have restored the ability to acquire Barnes maze. Now, we can also look at whether the animal can retain the ability retains the knowledge of where the target hole is. And so shown here is just an example of the target. And then what we do, so we train the animal for 4 days, and on the 5th day, we put the animal into the maze, and there's no target box. So the animal just simply starts searching for the hole, and there's no way to escape. And you can see here in the middle, you can see heat maps of the location of these mice during the probe trial. And you can see a sham animal readily investigates areas very, very close to the target. The saline animals do not know, obviously from this, do not know where the target is. And these are the animals that are treated with drugs first at 3 days after injury. And this data can be seen here in these scatter plots. This would be sham, this is injured, and this is injured treated with drugs. And so what it says is that there are a variety of targets that still can be targeted with drugs 3 days after injury. So we wanted to, since we knew something about the anatomical necessities to acquire these 2 tasks, we wanted to directly test the hypothesis that if we treat with drugs at 12 hours, we're improving both sides of the brain. And if we treat at 72 hours, we're improving things on only one side of the brain. And so I just remind you, when we hit the animal, we're hitting the animal laterally. So we'll call this the ipsilateral side, and we'll call this the contralateral side. And once again, you can see here the fact that you have these 2 structures here, are the 2 hippocampi of the mouse. And what we did was the way we could distinguish one hippocampus from the other is by doing classic hippocampal slice electrophysiology. And in this case, what we do is we take advantage of the fact that the hippocampus is a highly laminar structure. We can put into a stimulating electrode into a specific area of a hippocampal slice and know that we are stimulating the axons that go between the CA3 neurons and the CA1 neurons. And so what we can do then is look at the strength of the CA3-CA1 connection. And to summarize a lot of data, because again, it's a very short talk, I don't have the opportunity to show you everything. What we found was that our injury model Excuse me, I have to go back for one moment. And so what we can look at here is we can look at whether this synapse in fact strengthens. And the strengthening of that synapse is very well understood to underlie the ability to acquire either Barnes maze or active place avoidance. So we're basically asking the question, can this synapse be strengthened? And what we found was that injury bilaterally destroys the ability to strengthen synapses in a experimental paradigm called long-term potentiation, or LTP. And what we found is if we dose the drugs at 12 hours, we restored the active place avoidance, and we also restored LTP bilaterally. And if we treated at 72 hours, we would restore Barnes maze, and we only restored LTP in the contralateral hippocampus. And so this was a very surprising and interesting result to us, because what it says is that with the same drugs, different brain regions have different therapeutic time windows. And so our working hypothesis is that the brain regions that are most proximal to the site of injury, the pathophysiology occurs more rapidly. And in the areas where it is more distal, the pathophysiology occurs more slowly. And this is one of the things that we're testing in the laboratory right now. But one of the things we want to look at is why, in fact, we were restoring long-term potentiation. And I was very fortunate. To have a colleague of mine at Downstate, Dr. Todd Sachter. And Dr. Todd Sachter, Todd is an expert in the biochemistry of long-term potentiation. And so what Todd had shown is that when you stimulate the CA3-CA1 synapse, again, you release the neurotransmitter glutamate, and glutamate binds to two postsynaptic receptors, one called the NMDA receptor and the other called the AMPA receptor. And the thing that's most striking about the NMDA receptor is if stimulated properly, it will open and then flux the ion calcium into the neuron, into the postsynaptic neuron. And the thing about calcium that's most striking about as an ion is it's both a charge carrier as well as a second messenger. And there's a variety of calcium activated protein kinases in the postsynaptic cell. And activation of all of them, you need a very potent activation, releases a translational block. So the other thing that Todd had found is that in synapses is the RNA for a protein called protein kinase M zeta. And it is translationally inactive. And when calcium comes in through NMDA receptors, you can release that translational block. You can make PKM zeta. And this then, one of the things that it does is increases the density of AMPA receptors on the surface of the postsynaptic cell. And so this is a way that you get synaptic strengthening. And so we wanted to see if protein kinase M zeta was actually a drug target for our combination. And that is in fact what we found. So one can see here, this is immunocytochemistry, immunofluorescence of CA1 neurons that were treated with an antibody with PKM zeta. And these are sham animals, and you can see that there's ample expression. And you can see now in the saline animals, the expression of this enzyme disappears. And if we treat with the drugs at 72 hours, in the ipsilateral side, the drug levels do not return. And in the contralateral those, the drug level, the kinase levels return sufficiently for us to get LTP. And one of the things about this is this is a very persistent effect. So if we look at PKM zeta in sham animals and normalize it to sham at 100%, you can see that in injured animals, it goes away. And if we treat with drugs, it doesn't really change very much. And this is on the ipsilateral side. But on the contralateral side, once again, if untreated with drugs, even out to 2 months, there's no increase in the PKM zeta levels. But if we start treating with drugs at 3 days post-injury, we can maintain PKM zeta levels for a full 2 months after injury. And so what this is saying to us is that not only can we treat with the drugs days after injury, but we also can get persistent therapeutic effects. So one of the things that I'm working on right now very hard is to get this into clinical trials. And as I said, one of the things that's very important for a clinical trial is we have to have an objective measure of a therapeutic effect. And so one of the things that are being used, one of the things that's being used very commonly is to look at the release of brain proteins into the plasma after traumatic brain injury. And then we have now the ability, these very, very sensitive analytical techniques to measure the level of these proteins in blood. And so one of the proteins that we've been looking at, is a protein called tau, which is a protein, in fact, that's found largely in axons. And we can see the tau levels are relatively low in saline. And so we injured animals and looked at tau levels 1 day after injury. And if we treat with the sham animals have relatively low tau levels, the injured animals' tau goes significantly up. And if we treat with drugs first 12 hours, we can in fact lower tau levels, which suggest that we actually have an objective plasma biomarker for a clinical trial. And that's in fact what we're working on right now. So let me just summarize what I've talked about today. So I'm going to make the argument that TBI is going to be best treated with drug combinations than monotherapy. I think there are just simply too many things going wrong with the brain at the same time that we have to find combinations that work. And we can find, and these experiments are certainly suggesting that there are multiple drug targets that remain at least 3 days post-injury. And one of the things we're very interested in doing right now is really examining fully what the therapeutic time window of this drug combination in fact is. And the last thing, and then this was very much, a lot of this was driven by the fact that I teach pharmacology to medical students. And one of the things you learn is drugs do all kinds of things more than they were proposed to do. And so minocycline was used to treat bacterial meningitis and then found to be an anti-inflammatory. N-acetylcysteine is used to treat Tylenol overdose, but is now being used for a variety of different things. And so it says that FDA-approved drugs are very, very good potential resource for drugs to treat other diseases, and in this case, for a traumatic brain injury. And I've been, again, very, very fortunate to have a lot of very talented people in the laboratory who are working on this. I'd just like to acknowledge Dr. Elena Nikulina, who's a postdoc with me, Dr. Sangaboale and Dr. Whitney. Todd Sachter has been an invaluable collaborator in these, and my work with biomarkers is being done in collaboration with Kevin Wang and Sunny Yang at University of Florida. And I also like to acknowledge the groups that have funded this research. So thank you very much.
Thank you very much, Dr. Bergold. If we could get another round of applause. Wonderful presentation. It's really insightful to learn about your work. And last but not least, we have a presentation from Dr. Sacks-Zimmerman.
Okay, stop sharing. Okay.
Hi, everyone. Thank you so very much for having me. And thank you to everyone who voted. That's great. So I am going to share my screen. Okay. So this is a bit of a different perspective than the previous two speakers, and I think it's a sort of a good follow-up because I'm really talking about the more clinical manifestations of concussion, specifically the neuropsychological treatment of persistent post-concussive symptoms, PPS, and implications for return to functioning. So the purpose of this brief talk, and I'm going to try to make it brief, it's going to be hard. I'm going to outline some of the cognitive and emotional sequelae of concussion, explain the role of cognitive remediation and cognitive behavioral therapy in treating persistent post-concussive symptoms and addressing return to functioning in this context. So I think it's good to sort of define what we're talking about here, because TBI has a variety of different severities, as was previously mentioned. I'm really talking here about mild TBI or concussion, as defined by the Berlin Expert Consensus on Concussion. Concussion and TBI are very synonymous. Up to 90% of the brain injuries we see in the ED of the United States are mild. And mild TBIs are considered to be benign, rapid, short-lived, and there's a resolution in a sequential manner. However, long-term effects are often not benign. And that's what I'll be going into in terms of how we treat those folks. So the recovery after an uncomplicated concussion really follows a favorable course for the most part, within days to weeks. However, 10 to 15% really have persistent concussive symptoms. And that's who we focus on when we're dealing with neuropsych treatment. So what is persistent post-concussive symptoms? It's defined by this Berlin Expert Consensus. I should say it's a group of neurologists, psychiatrists, psychologists who came together to really define some of these more abstract terms when we're dealing with brain injury. So I keep referring to this Berlin Expert Consensus of 2016. They defined PPS as really this failure of normal clinical recovery in terms of symptoms that persist beyond the expected biophysiology of a brain injury, a mild brain injury. So that's anyone who still has symptoms greater than 10 to 14 days in an adult or greater than 4 weeks in a child. And they said that basically these persistent symptoms are likely due to coexisting or confounding factors, which do not necessarily reflect ongoing physiological injury to the brain. So with that in mind, how do we treat those? And again, it was illuminating to hear, all the sort of medicalized and pharmacological treatments and the research going on from Dr. Bergold. But as neuropsychologists, we're certainly interested in the census statement saying really biopsychosocial approach to the management of these chronic symptoms is standard of care at this point. So what is the biopsychosocial approach? It's this idea that we combine cognitive behavioral therapy or psychological treatment as well as cognitive remediation to treat these folks. So within the field of concussions, again, we're dealing with mild brain injury. There continues to be many unknowns and clinical dilemmas. I'm not going to go into all of them, except I'll say, as neuropsychologists doing treatment on these folks, we're really interested in the last two. So complications, or to what extent neurologic versus psychological factors contribute to persistent concussive symptoms. There's a growing body of literature that indicates psychological factors do play a significant role in symptom recovery and contribute to the risk for persistent symptoms. And we're interested in treatment, which approach works the best. And there's active research going on in terms of combination psychological treatments for persistent symptoms. So what are the risk factors for PPS? Again, there's many. A lot of research has focused on psychosocial, psychiatric, and neurocognitive factors that go into PPS. And as neuropsychologists, we're really interested in these pre-existing neurocognitive, psychiatric, and psychosocial factors. So these are some of the psychiatric factors, both predating the concussion and as a result of the concussion, that contribute to persistent concussive symptoms. Studies have found that those with persistent symptoms after concussion had more pre and post psychiatric difficulties and lower levels of functioning before the injury compared to those whose symptoms resolved within normal limits. Research that has looked at pre-injury somatization scores in high school and college athletes found that those with pre-injury somatic scores have prolonged concussion symptoms. So in terms of just the acute aftermath on neurocognitive functioning and concussion, attention, processing speed, memory and learning, and executive functioning are the main cognitive domains that are impacted. But we'll also see these domains impacted in this persisting level of functioning as well. What I should say is there's complicating factors that contribute to cognitive dysfunction in these areas, including fatigue and headaches, ongoing dizziness that can contribute to deficits in attention and processing speed as well. So this is interesting for us as neuropsychologists, because often when we see an individual, we do an intake where we get subjective self-report, and we're also doing objective neuropsych measures. And there's often a discrepancy in some of our concussion patients. So patients can self-report cognitive difficulties. That's a subjective measure. And they could do fine on some of our objective tests. So this is the discrepancy we're dealing with, and it does factor into the treatment we do. It's important to know, though, that perception of cognitive deficits, even if it doesn't show up on our objective measures, can still negatively impact one's sense of self and one's functioning. So again, I referenced this idea of a biopsychosocial approach to treatment is recommended in concussion, according to that consensus statement I referred to earlier. The two types of neuropsych treatment that I'm going to be talking about is CBT, which really addresses the more emotional difficulties, including anxiety and the impact of the injury on one's functioning, and cognitive remediation, which more addresses the cognitive difficulties after concussion, including difficulties with attention and concentration. Research has demonstrated that a combination of these two treatments really works best. So what you'll hear, or what I'll hear often when the individuals with persistent post-concussive symptoms walk into my office, is that they have difficulty focusing, they get easily distracted, they feel slowed down. And that's important because attention and processing speed deficits, if you sort of think about the brain as a pyramid, all our other cognitive functions are built on top of our ability to attend, process information, and the rate at which we process. So when those things are diminished, it impacts all cognitive functions, including our memory, our language, our executive functioning. It's also important to sort of think of how the emotional symptoms impact our cognition. So emotional distress is often very distracting, and it diminishes the bandwidth to which someone can attend and process information, and that can really diminish one's ability to return to learn, play, and life. So I'm going to start with talking about cognitive remediation. It's really a collection of techniques designed to improve cognitive difficulties, including processing of information, attention, memory, and executive functioning. It's really based on the concept of neuroplasticity. As we know, and was sort of referenced in the earlier speakers, that the brain is a plastic organ. It does have the capacity to change and alter neural pathways in response to injury. And some of the theory behind cognitive remediation is that it really expedites that sort of general process within the brain. Okay, so I'm going to briefly, I don't have time to go into all the sort of techniques and strategies we use with cognitive remediation, but attention process training is important to describe because that's often the cognitive remediation strategy that we start with. Attention training is really the practice standard for remediation of attention in post-acute rehab of TBI and concussion. It's made of exercises where we really teach individuals how to allocate and control their attention. It enhances awareness of their level of distraction. It's made up of repetitive drills with increasing attentional demands. The good thing about APT is that it really has exercises that target all different types of attention, including focused attention, sustained attention, alternating attention, and working memory. These are all factors of attention that go into our daily tasks, whether or not it's within an academic setting or vocational. This is a quick example of one of the working memory exercises of APT. I know it's just like kind of a bunch of numbers and what is this, except when you do this with a patient, basically they're required to start at a certain number and keep the above exercise as a more sustained working memory exercise where they keep those three calculations in their mind, +8, -6, +1, and they generate a whole sequence of numbers based on those calculations. It takes a lot of working memory load. Again, in cognitive remediation, we can simplify it so that the person really understands and enhances their ability to do this, and then we get to more complex exercises where they have to hold it in their head. The rule of thumb in cognitive remediation is we always sort of start with errorless learning, where we don't want any errors, so we support one's ability to do this exercise until they can do it with diminishing cues. The bottom exercise, we have the individual repeat a sequence of numbers. They have to hold that in their mind, and then they have to put it and manipulate these numbers into various orders. Again, it's a highly taxing exercise on working memory. But again, with support and cues, you'll really see that individuals can start to enhance their capacity for working memory doing these exercises repetitively. So what's the real life applicability of this attention training? We always focus on the components of attention that the individual is having difficulty with. As I mentioned, attention factors into those basic domains. And we really try to ask, what types of attention is impacting their daily functioning. And those are the exercises we'll choose to really help enhance one's functioning. Okay, so attention training, why do we start with that? Why is it important? So attention training really helps an individual to be able to hold on to information longer, to process it in a deeper way. And that really increases the likelihood of retention of information. I don't have time to get into it, but attention is basically married to short-term memory. You need attention to be able to enhance your ability to encode information into short-term memory storage. So attention training is essential before we work on our more short-term memory strategies. Attention training also helps an individual hold on to a step while performing it without distractions. So in that way, attention training helps with sequencing and carrying out of tasks without making an error. And so that gets into how attention training really supports executive functioning. So improved attention, which is really the goal of attention training, it helps control cognitive abilities and helps an individual have confidence in one's cognitive abilities. Okay, so now I'm going to talk about more of the treatment of the emotional aspects of PPS, and that's CBT, not to use too many acronyms, but CBT referring to cognitive behavioral therapy. So as you can see from this, there are many psychological factors that contribute to the emotional sequelae in these persistent concussive symptoms. Pre-injury psychological disorders, including anxiety and depression, as well as pre-injury personality styles, contribute to the tendency toward negative beliefs regarding cognitive functioning, which in turn leads to negative emotions. So some of what you'll hear when you're dealing with PPS in individuals is people feel sort of abulic, or they feel like they just don't care, they don't feel like themselves, they're afraid to do things, they're easily irritated. So individuals with depression or anxiety after concussion have a tendency for catastrophic thinking. They will always feel injured and have symptoms. Individuals with pre-existing somatization tend to have an external locus of control in their thinking, meaning they feel controlled by their symptoms instead of feeling they can function in spite of their symptoms. Individuals with overachieving personality style tend to engage in all or nothing thinking, where they must perform close to perfectly on a task or else they are impaired. So in CBT, we sort of tease apart what those thought patterns are. We examine those thought patterns and how there are resulting behaviors and emotions from those thought patterns, including avoidance and the emotions of self-doubt, helplessness, diminished motivation. The theory of CBT is that thought patterns really do contribute to one's actions and emotions. So with this theory, in concussion patients, we look at their thought patterns related to their sense of self, their symptoms, their injury, and their environment. And it's important to know that a lot of these thoughts lead to avoidance behavior, and we want to nip that in the bud to really help these individuals return to baseline functioning. Avoidance of activities also creates even more anticipatory anxiety because individuals are not letting themselves be exposed to the anxiety-provoking activity. So CBT increases awareness of thought patterns that contribute to maladaptive behaviors in the recovery. The treatment helps to reframe their thinking about their symptoms and the injury. As providers, we reality test their thinking and come up with alternative thoughts for various concepts. So for example, the treater helps to increase their awareness of stressors that might be unrelated to the injury. The treatment also helps them to change their thinking about re-engaging in tasks they have been avoiding. Like they can always stop an activity if they experience a symptom, and not every symptom is related to the concussion. And part of CBT is also behavioral modification. So behavioral modification that's addressed in CBT includes modifying work or class schedules so it's less overwhelming, using breaks during taxing cognitive tasks, as well as some relaxation techniques. And the whole goal of really reframing thinking and behavior modification is to change emotions. So to really alter emotions from helplessness or demoralization to more empowerment, more motivated, more confident, and feeling more in control. So much of what we do in terms of rehabilitation of post-concussive symptoms that are persisting is really the emphasis on the B, so behavioral reintegration, this idea of graduated return to vocational and academic functioning. So when a person has a concussion, they'll often see a neurologist. And there's no real roadmap for how do I get back to vocational functioning? How do I get back to my academic functioning? So as neuropsychologists, we really help them build that roadmap. And how do we do that? So this is sort of an example of a reintegration schedule in the acute period of a concussion. And you'll see it varies by the days post that we'll see an individual. So in the first 2 days after a concussion, rest becomes important, but we need to start to develop a reintegration roadmap thereafter. Any reintegration schedule, whether or not it's 3 days post, 8 days post, 9 days post, involves 4 elements. This idea, what you'll often see is individuals will stop going to classes, they'll stop going to work, they'll stop playing in sports. What we stress is this idea is that they must get to school or work, even if they don't stay. So that's one element. There has to be an element of physical exercise, an element of a cognitive exercise. That's something often we derive for them. It doesn't necessarily have to be an academic or vocational task. A sleep nap schedule. Again, often patients will just rest with no timer, or they'll rest until they feel okay. And we want to really discourage that. So we encourage, again, a sleep nap schedule that really sort of follows a requirement of certain time allotments. The patient is encouraged to utilize a daily planner to incorporate each of these 4 factors into time slots into their day. So the pattern of the reintegration schedule is that there's a gradual increase of each factor. So we'll gradually increase a cognitive task or a physical task, with the final goal being return to work or school with accommodations if necessary. And again, this will look different for everybody. So this is an example of a reintegration schedule that increases over time. So at Weill Cornell, we have a concussion clinic. It's virtual at this point, but basically it follows the following time frame. We're 2 neuropsychologists and a neurologist that really field a lot of these patients. The emphasis within the first 1 to 4 weeks is basically getting an objective neuropsychological data and self-report measures. For those that we see, and we see patients at different timeframes, because as Dr. Bergold mentioned, for mild concussion, people don't seek treatment until they're 12 weeks or more post-treatment. And for those that are 6 to 12 weeks post-treatment, the emphasis is often on assessment of pre-existing psychiatric factors and psychological and psychosocial stressors that might be maintaining some of these persistent symptoms. For those that are 12 weeks or over, the thing we emphasize in the intake is always the avoidance behavior. What are they avoiding that really is contributing to some of their ongoing symptoms and dysfunction? And for these folks, we always recommend an 8-week course of integrated CBT and cognitive remediation. So basically, patients are complex. Many have pre-injury cognitive and emotional difficulties. Even after optimal concussion outcomes, cognitive and emotional changes can occur. And neuropsychological treatment can help with both, independent of the origin. So the treatment of psychological aspects of PPS, we emphasize awareness and hope, awareness that there are some psychological issues on the perception of one's neurocognitive functioning. We discuss impact of ruminations in terms of attentional functioning, that concussion is an evolving picture, and that certainly change and improvement can always occur. We want to emphasize avoiding avoidance, so that's why we really focus on deriving an individualized reintegration schedule. And that's basically it. Again, I want to thank you, and I'm sorry it was so short, because so if there's any questions on the more specific aspects of the treatments, I encourage any questions after. So thank you so much.
Thank you very much, Dr. Sacks-Zimmerman. A big round of applause, please, from our audience. That was a wonderful, insightful presentation. Thank you very much once again. And now we would like to shift over to the question and answer portion of our event for the remaining amount of time. And so I'd like to reintroduce our events committee team member, Kosi, who's going to be moderating our Q&A. Just a friendly reminder, if you have any questions that popped up during the presentations or even right now, please directly message me. My account is currently called Columbia Synapse Chinmay, or message Ling Hao Kong, who earlier typed a message in the chat. So please send us a message with your question, as to avoid spamming the chat while our speakers are answering questions. Keeps the chat nice and clear. So thank you very much, and Kosi, passing it over to you now.
Hello, everyone. Good evening. Yes, thank you, Chinmay, for introducing me. So the first question goes to Dr. Koliatsos. It says, what led to you to work in your specific area of focus in TBI research?
All right. So a number of things. One is personal, actually. I had a very good friend I grew up with who had a fairly serious traumatic brain injury and who lived to see the chronic implications of TBI. Chronic TBI is a neuropsychiatric disease. What really burns the lives of these people is not so much that they cannot move or feel or talk, but they cannot emote, they cannot interact with others, they cannot work. So I saw that. The second is my formative studies were on Alzheimer's disease. I trained at Hopkins with Tom Price, a rather legendary figure in the neuropathology of Alzheimer's. And chronic frustrations on understanding and treating Alzheimer's. And the fact that I was using some injury models to understand some cellular and molecular processes led me think that research-wise, mechanistically, just because you have a clearly defined point of injury, point of start, to study mechanisms as they unfold over time would be a good way to look afresh into the problem of neurodegenerative disease. And as a matter of fact, TBI is now risk factors, not only for AD, but perhaps more so for Parkinson's disease and motor neuron disease, among others. So there's a personal and there is a research reason. And there's a third reason that's also very important. It's because I believe that as a neuropsychiatrist, and because I think chronic TBI is fundamentally neuropsychiatric disease for reasons I mentioned, it really grows me as a clinician. It allows me to think more in a piece of clinical work that I'm interested in very much so, which is interactions between brain and mind. This is my other half, really. I'm very interested in these two things. They're not the same. We have a tendency of equating the two. A lot of psychology is written in pseudo-color phrenological fMRI maps, but I don't think so. It's the complexity of this that actually is fascinating, and it certainly fascinates me.
Yes, I can definitely relate with that. And I think lots of people here can too. That was a very inspiring story. Dr. Bergold, would you like to tell us your own experience? What led you to your specific area of focus in TBI?
Mine is actually much less interesting in a way. It was actually unfortunate because in the mid, around 2006, 2007, we had this terrible problem with soldiers returning from Iraq who had, because of the change in way the military could protect people, they were all wearing body armor, but they all many of them had head injuries. And so there was a 1 year that the Congress allocated $700 million in 1 year for TBI research. And I thought that I had my background provided some valuable insights. And so I wrote a grant, and I had never studied TBI before. But what made me write the grant that I wrote was basically driven by two things. One is that I was teaching how to treat a large number of diseases. And so, for example, diseases such as cancer, such as HIV disease, such as tuberculosis, everything was being treated with combination therapy. And as I was teaching medicine in medical pharmacology for 20 years, it just seemed to be more and more and more. And the second thing actually comes very much from my postdoctoral training at Columbia, where I was working with two very skilled neuroscientists, Dr. Eric Kandel and Dr. Jimmy Schwartz. And we very much thought that the readout of the brain is behavior. Where, it's very interesting to look at all the various components of a nervous system, but ultimately what you want to do is improve behavior. And so that's why I developed this screening method to find drugs that actually improve the active place avoidance. And that's what got me. One was the combination, and the second was basically doing a very top-down approach where I said, okay, these drugs improve these animals' behavior. Now let's figure out what they're doing and understand something about them.
Yeah, that's amazing. I love the fact that you could connect your previous study to brain injury and all. It's really inspiring.
Well, let me just add one last thing, because I think it's critically important. Is when I was at Columbia, I was studying sea slugs. And sea slugs have a nervous system that is shockingly different in many ways from a vertebrate nervous system. But there are some basic tenets of neuroscience across evolutionary distance. And so many of these lessons I learned were from a much more simple system than the vertebrate nervous system.
Wow. Yeah, that's amazing. Sea slugs. Yeah. I would like to pass it on to Dr. Sacks-Zimmerman. Now, could you please share your experience? What led you to your specific research area?
Sure, and just a note, it's much more my sort of clinical practice than research specifically. So just because my sort of background is mostly clinically based, one word to kind of sum it up is, and I think Dr. Bergold is implying this, it's neuroplasticity, and it's absolutely an amazing function that comes so inherently and naturally to the brain. The brain can change, and the brain has enhanced capacity with exercise, whether or not you see that in a mouse model or working with human beings as we do in clinical practice. So, my road basically started with the postdoctoral fellowship in psychology. We get trained in very generally, and then your postdoctoral fellowships get you more specific in terms of the specialties that you become interested in. Mine started with geriatric psychology, where I was mostly assessing individuals who were mid to late stages in neurodegenerative and dementing conditions. And while it was fascinating to be able to have these tools to measure cognitive functioning, there was not much you could do in terms of interventional neuropsychology, where you could use strategies to help enhance functioning, because it was just the nature of the disease. Individuals get worse, unfortunately. But nonetheless, the working with families and really being able to support one's functioning was quite satisfying. But it led me to my second postdoctoral fellowship, which was in specifically working with traumatic brain injury patients and looking at research on metacognitive strategies. I didn't talk much about that, but how metacognitive strategies, as well as those restorative drills that I talked about with attention training, can really enhance one's ability to process information and to use strategies to help them optimize functioning. So once I started working with the TBI population and I saw the effectiveness of a lot of these strategies, the roads I took in terms of career led me to continue using these strategies, but with a variety of different patient populations that suffered from cognitive issues, including individuals that had cognitive dysfunction post-chemotherapy treatment in cancer patients, individuals with different neurological diseases, including MS. And currently my position at Cornell, I work for the majority of the patients I see are these post-operative neurosurgical patients that have a variety of different types of diagnoses, and using these cognitive remediation strategies with such a variety of populations and seeing how people can improve with the support that neuropsychologists can give them is just where I am today, and the sort of defining feature of what's led me through the path in my history.
Yes, I agree. It can be really daunting to see people just getting worse and worse. But like this idea of neuroplasticity, it's amazing. Like really, the idea that the brain can change, and I believe it affects a lot of different areas of our lives, not just the medical aspect. Yeah. So thank you very much for sharing your experiences. So Shimai wants to make a quick announcement. So I'll let her go.
Thank you, Kosi. Hello, everyone. Again, it is currently 4:33 p.m. And so we formally scheduled our event to go until 4:30 p.m. So our speakers, are you by chance able to stay back for a few more minutes, or otherwise we can also conclude either way?
I can certainly stay on.
Awesome, thank you.
as well.
Thank you. We'd love to continue with a couple more questions and go over a little bit of time. And our audience members, if you are unable to stay for this go over time period, please feel free to email us questions afterwards as well. And we can try to follow up with our speakers and see if we can answer your questions as well. I will send over the Synapse email in the chat at the moment. So please feel free to email us if you have any questions, and please look out for the video recording of this event to be posted on our YouTube channel as well. And I believe we'll be sending up a follow-up email with that announcement as well. So thank you, everyone. If you need to leave, thank you so much for joining us today. And Kosi, back over to you.
Thank you, Shemaim. Okay, so my next question is directed towards Dr. Koliatsos once more. So would you please tell us a bit more about the connection between TBI and neurodegenerative disorders? Have you found any definite connections between the neurodegenerative dementias and previous TBI occurrences in older adults?
Well, if you're asking me if I have found, I guess some of my work points to that, much like Peter and others. There are several well-defined pathways. One of them is clearly based on my previous work, which is that if you cut axons, you have a number of processes that have to do with transsynaptic degeneration. Remember, I showed you the corpus callosum of this person who had diffuse axonal injury. The thinning out of corpus callosum is actually a very strong biomarker that the interhemispheric connectivity is gone. There is a very well described cellular and molecular pathways via which cell bodies whose axons are cut die retrogradely, and also cell bodies that receive the input from an axon also die transneuronally. So one is transneuronal degenerative havoc. Another is chronic neuroinflammation that, again, Peter alluded to, although I'm personally not actually as convinced that it is definitively a cause, perhaps because it's very difficult to describe as a biological phenomenon. The world of inflammation is extremely kaleidoscopic. You can think that you have a bad microglial cell, that it's bad now, but in a couple of hours may turn to somebody else. So I think I have a problem I have a problem in the scientific method there. So I'm not sure about that, but this is actually a mechanism that's out there. And third is proteinopathy. Again, Peter mentioned tau. The idea here being that perhaps through actually axons becoming jeopardized and impaired, there's certain ways by which proteins that are normal proteins in cells start behaving like particles, like prions. And nerve cells don't like that. What's worse, these particular proteins, or actually aggregated proteins, go from synapse to synapse, and as they travel around the nervous system, cell bodies or neurons, neuronal cell bodies die. So we have this proteopathic sort of pathway which may lead from injury by jeopardizing axons. For example, axons may get leaky, or axons may signal otherwise, or actually disruption of the blood-brain barrier that exposes these normal proteins to serum-borne molecules that chop them up in ways that are not native to the nervous system. They can actually lead to prion-like mechanisms. So these are at least three, or if you include brain barrier disruption, 4 mechanisms by which we can go from the acute event to a degenerative disease. And this is exactly why TBI, besides an important issue in and of itself, it is also a very useful, in quotes, entry point to study what might happen in a neurodegenerative process that you would otherwise not be able to, because you do not have a time definition of when Parkinson's or Alzheimer's or frontotemporal degeneration of Lou Gehrig's disease begins.
Thank you for sharing, Doctor. Hopping back on, as Kosi unfortunately had to step off due to a power blackout, Zoom technicalities and power difficulties. But I think she's hopping back on right now. But in the meantime, I'd like to introduce our final question. This is open for anyone. This is a question from our audience member. For those who don't seek treatment for their traumatic brain injury until months or years post-injury, what does your research or clinical work show about recovery? And Dr. Bergold, would you like to start off with this question?
I think that's a fascinating question. I think it's extremely important. And one way to think of it in some ways is just historical. I mean, neurology 20 years ago was once described as diagnose and adios. I mean, they could make it, there was relatively little one could do to the patients. And now it's very clear that the brain is much more plastic and much more ability to repair than before. And I think we're simply just beginning to explore this in a deep way. I mean, one of the things that we're trying, with the help of the VA, is to start, dosing drugs, months after injury and see if we can in fact get therapeutic effects. The other thing, of course, which very much segues to the other speakers, is clearly cognitive and psychological approaches work, and our hope is that we can augment them, in fact, with drugs at the same time. So I think that this is a challenging problem, but I think 10 years ago, 20 years ago, people wouldn't even consider this a problem at all. They would simply say you can't do it, and so I think that the future looks hopeful for that very reason. And I think this is going to be true of a variety of neurological diseases.
Thank you very much for sharing. Dr. Sacks-Zimmerman, would you like to comment?
Yeah, I mean, I definitely agree with the hopeful answer of Dr. Bergold. I mean, what I'll say is just the first sort of phrase that comes to mind is the patient role and how people sort of conceptualize their identities after an injury, whether or not it's mild in the concussion range or more moderate to severe. And to some extent that can be adaptive, right? Because awareness for cognitive and emotional changes can help people compensate for them. And seek treatment, et cetera. But on the other hand, if there's sort of an overabundance of identification with the patient role, it can also lead to that avoidance cycle I was sort of talking about. And in our treatment, again, we work on behavioral reintegration because we really want to kind of nip that in the bud in terms of how avoidance starts to lead into that patient role. So I'll see patients sometimes who are 10 years after an injury, 5 years after an injury. And of course, if you look at a span of 5 years in anyone's life, whether or not they're 20, 30, 40, so much can happen in terms of contributing to cognitive dysfunction and emotional difficulties, but also that it just might not be exactly related to that injury. And yet so much of what they attribute to in their narrative of their sense of self goes back to that injury. So the answer is yes. I mean, you can treat someone years later, but you want to be able to conceptualize all the complexities that are time and an individual progressing through time in their life in order to be able to address all those factors that might contribute to dysfunction. And to Dr. Bergold's point, if there's medication that can support that journey in terms of the treatment that they get, it's a great adjunct treatment to neuropsych treatment as well. So yes, there is certainly hope for individuals who are years post-injury.
So I would agree that it would take medical and non-medical, well, I shouldn't really say that, I don't like the term, psychological and non-psychological approaches to treatment for all degrees of severity. In my clinical experience with TBI patients, you can have the same contusion in the frontal lobe of two people. What kind of person patient A was prior to TBI, including genetics, habits, personality, other exposures, and so on, substance abuse, has a lot to do with the outcome of that same contusion. So at all degrees of severity, that's very important. But also you need a cadre of clinicians being involved in a supportive family. You need community. We need communities. We need communities. And one thing that suffers the most, at least in my experience, we don't have communities very often in these patients. Families discouraged. We don't have enough rehab programs. We don't have enough experts at any level, from social worker all the way to neuropsychiatrists and neuropsychologists. We don't have many. So this is why we need to really work some more to build communities. You cannot treat severely ill chronic patients without having communities. So that's something that we need to really keep talking about. And the other thing I wanted to say, at least conceptually, I think I would view treatments as treatments that may be preventative, treatments that may be just immediate post-exposure, and treatments that could be chronic. For example, I gave you 2 examples of MAPK inhibition that might be preventative. So you have a football game and you take a pill. Well, that's obviously a dream. That's a fantasy, rather, not even a dream. And then you have a post-exposure sort of prophylaxis, to use a term that applies in other problems in medicine. For example, like a NAMPT inhibitor. Because we're talking about time windows are very, very important. So importing models from basic science or translational science into what we do, and keep it in mind that it would have to be sort of the overall guidance to this problem.
Thank you for sharing your insight. It was really wonderful to hear your perspectives on recovery and time frame as well. And as a final note, before I pass it over to Sharik, our president, for closing remarks very quickly, I would like to mention once again, if you have any questions that are unanswered, please feel free to email us. I dropped the email in the chat. And thank you once again. And with that, I will pass it over to Sharik.
Great, thank you, Chinmay. Thanks so much. And thank you so much to all the speakers that joined us today, Dr. Bergold, Dr. Koliatsos, and Dr. Sacks-Zimmerman. Really appreciate your time and like kind of sharing your knowledge with the large audience. Like we said, Columbia Synapse, like at the helm for our mission is really raising awareness about brain injuries. So like a social and neuroscientific understanding of how brain injury manifests. And our population, it's a silent pandemic, and it affects a lot of people globally, but in the United States too, and affects our healthcare system in ways that, of course, like we didn't get into in this talk, but like we hope to in like future presentations as we understand the public health implications of traumatic brain injury. But with having said that, though, I just want to thank everyone who was able to join us today. And I know a lot of people were not able to stay a bit over, but for the people who were, thank you so much for staying over and listening to some of these great answers and to the insights that our speakers shared with us. And also I would encourage you to sign up for our mailing list and to look out for future events, as we hope to host in the future semester, the spring semester, and a conference. And we look forward to staying in touch with you. Thank you.
Thank you.
Thank you very much.
Thank you.
Nice seeing you, Peter.
Yeah, very nice seeing you too. And thank you very much, because one of the things that I've always learned is everyone has a different take on this, and everyone has a lot to teach each other. And I really enjoyed everything I heard today.
Me as well. Thank you so
Ito.
much.
Okay.
Thank you. Thank you.
Sharik's back-to-back meeting.
Yeah. Hi, Christina.